Today we're going to start looking at blood vessels and how they work, the different kinds of blood vessels. So if we look at types first, blood vessels, there's arteries, arterioles, capillaries, venules, and veins. All of these are a closed circuit, which means your blood stays in these vessels. The blood does not leave the vessels. Blood can be carried away from the heart to your cells where nutrients are taken, waste leave, and then they go back to the heart again. So if we look at the artery and arteriole section, arteries are very strong, elastic vessels. They're adapted for carrying high pressure blood. They carry blood away from the heart when blood is leaving. So that strong squeeze from the heart creates high pressure. So these vessels typically are round and strong and they hold their shape whether they're full of blood or not. As our blood vessels branch, they just go kind of like a tree. They go from a big branch into smaller branches. So as arteries become smaller, their name is actually given arterioles. So arterioles are just smaller branched arteries. Tunica, I know some of these, I'll have diagrams of these on your test. So on your cadaver lab or your cadaver website, make sure you look at some of these tunicas. An artery has an endo, remember endo is inside. So tunica interna, or internal, inside. Tunica media, which is the middle, made out of smooth muscle. And tunica externa, which is the outside. On your test, you'll have diagrams. So kind of know the difference between arteries and veins. I probably won't ask like smooth muscle. I will not ask what type of muscle makes up the tunica media, but I want you to be able to find where it is located. So the endothelium or inside, tunica interna, simple squamous, it's very thin, it creates a smooth surface so that blood doesn't catch and build up, gives off chemicals that prevents clotting, and basically helps entirely with blood flow. The job of the tunica interna is to make sure that blood flows easily. The tunica media in an artery is very thick. This is the part that gives it structure, and on your pictures you should see the thickness of that middle layer. The externa tunica then attaches to the artery. It's the outside layer, and it attaches to surrounding tissues. Two terms, vaso deals with vessel. So vasoconstriction is when it tightens. Vasodilation is when it expands. So arteries can constrict as directed by sympathetic impulses. When those impulses are inhibited, those blood vessels will dilate and get larger. And that size of that tube regulates blood flow and it regulates blood pressure. If I have a tiny little opening, it's going to have higher pressure because a lot of blood is flowing through a small area. When my vessels dilate and get larger, the blood flows through easier, which will lower the blood pressure. And then as my arteries go from large arteries to tiny capillaries, the walls get thinner because at the capillary area, that's where stuff is exchanged, and the walls have to be very thin. So here's a good comparison. I like this diagram. So the lumen is the opening in the middle, so that's where the blood flows through the center. Veins have valves. We'll talk about why later. Veins have valves, arteries do not. If I look at my inside layers, they are about the same for an artery and a vein. Connective tissue about the same. This middle layer, a lot thicker in an artery, and that again deals with blood pressure, the pressure of the blood going through it. And the tunica media in a vein is much smaller. External tunica, again, pretty much the same for both. So big difference. Tunica media is bigger, thicker in an artery, and veins are the only blood vessels that have valves. So in these arteries and arterioles, there's going to be muscles that wrap around it that help to squeeze or pulse the blood through. And I can see here I'm in an arteriole, smaller branch is a capillary, and then a sphincter muscle is right around the capillary. That diagram probably not as important. Capillaries are the smallest vessels, so they're kind of the in-between of the artery and the vein. They consist of a layer of endothelium. It has to be thin so that substances can be exchanged. I need oxygen to travel through, carbon dioxide, nutrients. So that permeability of the capillary varies from one tissue to the next, depending on what cells they're next to, what those cells need. Examples, your liver, intestines are going to have more permeability. Thinking of your intestines, that's where all your nutrients go through. Things are going to go through them much easier. So some organs are more permeable than others. Here you can see in a capillary, there is actually fluid that can leak through. Stuff can enter and leave this single layer of the capillary. And that is where exchange actually happens. In a real micrograph, so actual underneath a microscope, here I have an arteriole, here I have a venule, so small vessels. Capillaries are in the middle, and I can see where, all right, my red has oxygen, and it has nutrients. In these capillaries, that oxygen is taken up by the cells, and then my blood turns to blue, and then it's going to go back through the venule. So blood is coming this way, coming out, coming out, coming out. Nutrients are exchanged, waste are put in, goes to the blue, and this is going to go back to the heart. So that's how the exchange takes place. Pattern varies greatly. For example, muscles need a lot of oxygen. They're going to have high areas of capillaries. Your lungs have high areas of capillaries. Precapillary sphincters are just little muscles that basically regulate how much blood is going into those capillaries. Blood that enters the capillaries have high concentrations of oxygen. They're carrying the nutrients that is in the blood, and then that diffuses out of the capillary into tissues and cells. It's used by the surrounding tissues. Plasma proteins are going to stay in the blood because they are too big, so they don't enter and leave. They stay inside of those capillaries. A couple of small things. Hydrostatic pressure drives the passage of fluids and very small molecules back and forth at the arterial end. At the venule end, this one probably a little bit more important because your veins, the blood's not being pushed through, it's coming back. So osmotic pressure is what causes a lot of the fluid to return to the bloodstream in a vein. Lymphatic tissue or vessels exchange excess tissue fluid and put it back into circulation. A little diagram, not going to point much out here, but you can kind of see if I have an artery, there's a lot of outside force. This is why this has to be so thick. If I look at my veins, they're going to be thinner. There's not as much outward force. So in an animal or a human, your arteries, that thick middle layer, allows your arteries to keep their shape. Veins, if veins don't have blood in them, they will become flat because they don't have enough muscle to help them hold their shape. And I'll stop there for a moment.